Okay, so in this video I'm going to explain you how to perform a strict plate technique. In this technique, what we're trying to do is to separate two types of bacteria from a mixed culture. That's the purpose of the strict plate technique. And just as for any work in microbiology, you have to use the aseptic technique, which is sterilizing your loop in the middle of the flame of the Bunsen burner, 45-degree angle, until it's red hot, and then let it cool for a minute so you can prevent bacterial contamination, killing bacteria with a high temperature of your loop. And in this case we have only a single type of bacteria, but ideally would be to have a mixed culture. Now this is S. epidermidis, and what I'm doing right now while I wait for the loop to cool is to shake this culture, because the bacteria tends to sink into the bottom of the test tube by gravity. And of course, if I want to grab bacteria, I don't want to introduce my loop towards the bottom of the test tube. I only introduce it toward the half, and I want to have bacteria in the middle of the or the half of the volume of the test tube. Now, then one thing that you will do is, of course, having your label for the strict plate technique, your initials, and the date. And you will imaginarily subdivide your Petri plate into segments or regions. So you will start on one edge by putting your inoculum from the bacteria inside the surface, of course, of the agar, and then it starts touching the surface with the bacteria surface of the Petri plate, and then go sideways like this, like 4 to 5 times, and then in the area where you end, just take away your loop, sterilize it again, and then let it cool, and then streak 4 to 5 times, touching the edge where you previously end, and then any one in the opposite end. And then you will sterilize your loop, let it cool, streak again, sterilize your loop, let it cool, and every time that you're doing that, you're supposed to touch the area where you have the bacteria before. And then once you end on that area, you will go into the center, not touching the previous regions where you have inoculated your bacteria. The purpose is that with every streak that you do, you're supposed to have less and less amount of bacteria. So again, let me keep checking these to have more bacteria, and then grab your inoculum, close your tube, put it on the rack, open next to the sterile area your Petri plate like this, and start putting your bacteria. And don't lift your your loop, and then that's where I will end. Sterilize, let it cool, and then kind of keep an eye of where you end. So I end up like in here. So you have to create overlapping patterns. And when you are sterilizing your loop, every time between the strict regions is to dilute the amount of bacteria that you're dragging. You are doing a physical dragging of bacteria. So I'm kind of done in here, and you can see, even if you want to see through the light, you can see your streak where you end. So I think it's cool enough. So I'm going to grab bacteria from the previous streak, go sideways like this, and then end in there. in here, and I put even my finger as a reference outside, of course, the Petri plate. Never touch the Petri plate inside. Sterilize your loop, let it cool, and this takes patience with the sterilizing of every time, but if you don't do that, you're going to grab a lot of bacteria. And then some books, they show you, like their textbook shows only like 3 streaks, but sometimes people can make up to a pentagon and then go to the center without touching the areas where you previously streaked, because that's where you have more bacteria. So now I'm going to rotate a little bit the Petri plate, and then see, so this area where I have my index outside, that's where I end, and then here my middle finger, I will have it in the area where I'm going to kind of use as a reference to end my streak. So you touch then from here, from the edge of the Petri plate, going like this, zigzag, and then in here I end, where I have my middle. And then sterilize the loop, let it cool, and this is going to be for me my last streak. I did 3, and then I'm going to do the final one towards the center, but you can do a 4th one. And I will start where I have my middle finger, because that's where I end, and I have to, of course, touch part of the previous streak. Otherwise, I won't drag any bacteria. So I think the loop is cool enough. So I will touch the area where I had the previous streak, go sideways like this a little bit, and then towards the center and up to there. So I'm not touching the previous streaks. Sterilize your loop, and then you're done with the loop. Put it into the area where you have them. Close your Petri plate, and then remember to incubate your Petri plates. The lid, which is the wider part that covers the base, goes to the bottom. Why? Because you have media here. The media has water, and with the temperature of the incubator, this water from the media will evaporate, goes into the lid, and then it will condensate like if it was raindrops, and then it will go into the surface of your Petri plate, and it will make this bacteria that is growing in the surface of the Petri plate to be smeared all over the media. So you don't want that. So you will take it like this, close both lid and the base, and put it in the incubator at 37 degrees Celsius, 24 hours. See, the lid is going here into the bottom. And that's all for this video. See you in the next.